Good morning, everyone. And it's great to be back. I've been away for some time, company revamping and stuff, rebranding. So it's great to be back in the amphitheater with all the Science Circle friends. So, what I'm going to do today is this is not a conventional research presentation. I would classify this part of my research life into what I call the attic science. These are ideas I get, I present to the scientific community. Of course, I did this when I was a professor at Western Kentucky University in the biology department. That was 3 years back. Spent around 13 years there. But this was one that never excited everyone as I hoped it would. So it went into my attic. But today I thought I'll let it out again to see whether somebody amongst the science circle would like to look at it as a story. Okay. So yeah, it might make sense, it might not make sense, but that's all science is all about, right? You just give some ideas out. So let's get on with that. Bamboo, basil, and rice. When I first put this slide up, somebody said it's amazingly it's an amazing topic, but it also reminds them of a stir fry. I said it is a stir fry, but the stir fry as in it'll definitely stir your brains. Okay. Technically, I want to propose that transgenic plants can be non-human models to explore molecular basis of cancer and other inflammatory diseases. But I don't want to lay out and get into the topic yet because I want to give a premise. I want to give my background as a researcher briefly before I get into the main topic. So let's get on with it. The first thing is, what's the premise of my talk? Where am I getting this kind of ideas? The premise of my talk is right there in the rotunda of the University of Notre Dame. So there you see the legendary Theodosius Dobzhansky, who said nothing in biology makes sense except in the light of evolution. And this is something which I lived by because I read this paper. It was a paper which was published in the American Biology Teacher. That was a journal which sadly not many of us read, but that's a journal where I saw this 4-page paper which said, the title itself said, nothing in biology makes sense except in the light of evolution. It was not a conventionally written paper because it starts off with the story as recently as 1966. This was written in 1973 when Sheikh Abdul Aziz bin Baz asked the king of Saudi Arabia to suppress a heresy which said that the sun is running in the orbit and earth is fixed and stable. And he goes on. But the beautiful thing in the paper is he talks about this enzyme called cytochrome C that plays an important role in the metabolism of aerobic cells. It is found in the most diverse organism, he says, Dr. Dobzhansky, from man to molds. And he puts a table and he says there are different orders of mammals and birds and is looking at the amino acid differences. There you see, cytochrome C in a human has just one amino acid difference from a monkey. 13 in dogs, 17 in horse, and he goes on and on and on. Rabbits, kangaroos, ducks, chickens, fish, and finally yeast. 56 amino acids. But he says that's what it's all about, right? Nothing in biology makes sense except in the light of evolution. You will see how this influenced my idea, right? Now let's get to the topic. What are we going to learn today? Is there more to this thing called acetylcholine than its neurotransmitter abilities? What is acetylcholine doing in places outside of brain? We think acetylcholine is just in the brain, but it's outside the brain too. It runs through our body, what I call the non-neuronal acetylcholine. And the biggest thing I learned was it's in plants. So, can plants be non-human models to answer questions related to molecular basis of diseases involving inflammation? Where am I getting this weird idea, right? Let me step back a little. Like that paper I mentioned, which changed my life, this is a textbook which changed my life. I never looked at textbooks. Every time since I was an undergrad, we used to buy these bulky textbooks, which we never read, and we go back to the second-hand bookshop to give back and get our $25 back on $100, right? But this was a textbook I kept. This was a textbook, the first edition actually. When I was an undergrad, way back in '88, I bought this book called The Molecular Biology of the Gene by James D. Watson. The subsequent editions don't do justice to the first edition. This textbook read like a storybook. And in that, I found the definition of a gene, which I kind of memorized, right? How does Jim Watson, there you see him on the Macintosh commercial, which says, think different, right? I had to put that up poster in. Also, think different, because that was the series of ads in Macintosh which said, here's to the crazy ones. He's one of those crazy ones. And I inherited that crazy stuff from him, right? But what did he call a gene? He says gene is a discrete chromosomal region responsible for a specific cellular product, and it consists of a linear collection of potentially mutable units or mutable sites, each of which can exist in several alternative forms and between which crossing over can occur. I've never seen a more beautiful definition of a gene, which talks of a piece of DNA, right, which can be existing in several alternative forms. So that's what it's all about. So this is where I began my life as a molecular biologist, because textbook changed my life. Later on, I became a genetic engineer, as they call it, and that's when I came across defining terms before I go ahead, right? So what is transgenesis, which is also called genetic engineering? Process of transgenesis, because we're going to talk about some transgenic plants as models. The process of transgenesis means you are introducing desirable genes, transgenes, you're transferring genes into crops and ensuring their stable expression in the resulting offspring. So it's stable integration, a desired levels of expression, predictable inheritance. So you could be controlling that. So when I was a doctoral researcher, I constructed gene libraries. So I took those pieces of discrete chromosomal fragments and made a library out of it, made binding folders to isolate a gene called fatty acid desaturase in peanuts, right? My fellowship used to be like $100 a month. So I used to have a poster behind my working bench saying, I'm proud of working for peanuts. And then my advisor is like, are you talking about the crop or are you talking about your fellowship? That's how I started my life, working for peanuts, right? And when I became a postdoc, I worked with sorghum, one of the most difficult crops, where I introduced transgenes, but the plant punched me right back. All of my transgenes were silenced, which I had to reactivate. Then I worked with cotton, I worked with rice, and then I subsequently worked with bean. I became a plants guy, okay? Basically, that's what you're looking at here is a herbicide resistance gene I put in sorghum. When I spray herbicide on the normal sorghum, everything dies, and when I spray the same herbicide, the plants are happy. So this is one of my first photographs, which I've proudly displayed when I first became a genetic engineer. And then I did what is called as an environmentally friendly fungal resistant cotton. Basically what I did was, cotton farmers used to spray Trichoderma virens, a fungus antifungal agent, a biocontrol agent on cotton, because the trichoderma contains chitinase, which dissolves chitin of fungus. What I did was I took out that chitinase gene and I engineered it into cotton. And since I didn't use a synthetic gene or a gene from another plant, I used a biocontrol agent. I marketed this as environmentally friendly fungal resistant cotton. But that's another story some day I'll tell you. It's a very interesting story. But what I'm trying to tell you is I'm delving deeper and deeper into the DNA world of the plants, right? And then I worked with specific promoters. I can promote the expression of a gene right down to, as I can see here, as I am showing you here, a protoxylem of cells. And that's almost a decade's worth of postdoctoral research, which I just shared in 3 different slides, right? Now, I became a assistant professor at Western Kentucky University, and they said that, okay, we are going to take you to a research incubator where you would design your lab and you would take your classes there because the students will be exposed to other startup companies within the incubator. I was all excited. I landed and they took me to a tobacco warehouse. And they said, this is where you will develop your lab. You see on the left, that's what I was presented with. A 220 square feet old tobacco warehouse space, and they said, yeah, make your lab here. To my credit, within a month, I made that space like this. And this is where my story starts, what I'm trying to tell you today. Because I already worked with sorghum, I said, why don't I engineer sorghum with cellulase genes from a bacteria called Acidothermus celluliticus. What it does is it breaks the cellulose in the plants. My hope was I take these sorghum varieties which grow almost 7 feet, and I said, if you want to make biofuels from corn and soybean or whatever, you're cutting into the food supply, whereas sorghum is mostly a cattle feed over here in the United States, though it's a very staple crop in the East. I said I can use those thick stalks of sorghum. I put in the cellulase gene inside. I extract this cellulose, and the cellulase has already broken them down, and then you can have fermentable sugars for biofuels. So I presented this idea, got a small startup grant, both from NSF and my own university, and I was working. But this is where it is. My greatest fortune was I was in a university which had 75% teaching and 25% research. So that's where it is. I go into a classroom, sometimes 300 students, sometimes 25 students, sometimes 50 students. When one of my students, he's now a chiropractor in Texas, his name is Joey Ginto. He said, why are you working with sorghum? And you said it's tall and it's 7 feet. Why don't you work with bamboo? Then I said, wow. Because he said bamboo is an important multi-purpose plant grown in the world, raw product for ornamental, building, food, medicinal uses. Why can't it be a potential bioenergy feedstock? Because it can also sequester atmospheric carbon dioxide and help fight climate change, right? Now, that's what I thought. I would go and look at bamboo, right? But when I Googled bamboo, I came across a very surprising paper. It said, acetylcholine, evolutional study. What is this? And then the abstract itself just made me jump in my seat. Acetylcholine is a well-known neurotransmitter in the cholinergic nervous systems of vertebrates and insects. Very indirect evidence in lower invertebrates and plants and fungi. So this group, this unassuming group from Japan, it investigated plants, Arabidopsis, eggplant, bamboo shoot, cedar, hinoki, pine, podcarp, fern, horsetail, moss, yeast and mushroom, bacteria. They just assayed acetylcholine and they found that in the upper portion of bamboo shoots, there is 2.9 micromoles per gram of acetylcholine. That's like, I'll show it in a subsequent slide, it's 80 times acetylcholine than what is found in the brain of a rat. I was shocked. That's when I delved into what is called the non-neuronal acetylcholine. Yes, they don't have a nervous system. The plants don't have a nervous system. What is acetylcholine doing there? But It's not just plants. We have a non-neuronal acetylcholine flowing through our body, not the neurotransmitter, the non-neuronal acetylcholine. It's implicated in the regulation of cell differentiation. In plants, phytochrome-mediated processes. That's when I jumped in my seat again, cytochrome. What Theodosius Dobzhansky found in every realm of life. Non-neuronal acetylcholine also is implicated in cytoskeletal organization, immune function, ion transport. I didn't sleep that night because the paper also said there is a clinical significance of the non-neuronal acetylcholine. If you look at it, since it's involved in cell differentiation, every disease from the most mundane to the most dreaded disease like cancer or Alzheimer's, it starts with acute and chronic inflammation, a local and systemic infection. And then they say this non-neuronal acetylcholine levels are what causes, or the perturbation rather, causes dementia, atherosclerosis, cancer. And I said, now is the time I have to do a Dobzhansky treatment to this particular compound. So there you go. We started walking on two legs and we started making tools. We started making defense weapons, and then we got down to cleaning the place with a mop. Then we started playing golf. But ultimately we had to go back on all fours in front of this intelligent machine, and in comes bioinformatics, right? So the molecular evolution of acetylcholine, should we look at it? Can we uncover its transition from that non-neuronal role to the neuronal role? Was there a transition in that evolution? I'm still digging for that answer. And I was just an infant in bioinformatics at that time. I never took a formal course in bioinformatics, but I ended up teaching one of the first courses of bioinformatics in my university. I started my life with this book called Bioinformatics for Dummies. That was written by a great guy, Cedric Notre Dame. He was the guy who invented a program called Tea Coffee. I mean, you should just Google Cedric Notre Dame and tea coffee, and you will see how he came about writing that book. So I taught myself bioinformatics, and then I went into the classroom to teach. And one of my first exercise with the student was evolution of the seed storage protein called phaseolin. And when I was looking at those domains of phaseolin, the most ancient ancestor was microscylla. It jumped from plants to bacteria to microorganisms. So I said, okay, I'm no stranger to this kind of questions, right? So I said, okay, let's look at acetylcholine. There are two enzymes. One is the choline acetyltransferase, chat, as we call it, right? And I'm just amused. Now we have a chat GPT. So chat is not chatting. It's actually choline acetyltransferase. Maybe that's why, I don't know. Maybe there was a neurobiologist who was involved in that name, right? Choline acetyltransferase is the enzyme that synthesizes acetylcholine from acetyl-CoA and choline, right? Acetylcholine esterase was the one which actually degrades that acetylcholine so that it is usable. Now let's see. What did I do? I said, let's blast the chat. And when I blasted chat, I had this piece of paper stuck on my PC. It's a page from Charles Darwin. It says, I think, and he draws a phylogenetic tree, right? Let's see. Wow. Where am I going with this? Am I going to retrace some steps which have not been traced before? So acetylcholine pathway evolution, it is starting with blasting the chat. And this is a very refined tree, but it tells the story. It starts with us, then it goes to a shrew, then it goes to the naked mole rat. These are chat sequences. Then comes our Stuart Little, Rattus norvegicus, or the rodent, and then the pig, the bat, Colombia, the pigeon, then the chicken, and then the zebrafish. Wow, a model organism. I said, wow, now we are talking. The inner fish, as Neil Shubin would have us know, we all have an inner fish. So I was not still surprised. I mean, this is going good. The evolution is going good, right? Well, BLAST is a program which we use, a basic algorithm where you take a protein sequence or a DNA sequence and put it into this program called BLAST, and it just fishes out related sequences. So that's what I just did that BLAST the chat. So yeah, it's of course it's violent in its own way, or non-violent, as you might want to look at it. So so let's go on now. Okay, till now the tree looks fantastic. It now goes to Siona intestinalis, and now we're going to lower organisms, Digesia japonica, all fancy sounding names. Then we get to the insects. Still, I'm not surprised. We get to C. elegans, another model organism. Great. Trypanosoma. And that's when I stop in my tracks. When I see this entry, Ectocarpus siliculosus, and I'm like, no, that doesn't look like anything I knew before. And it's actually brown algae. Oh my God. Now I said I leave BLAST and I go to TARE, the Arabidopsis database. And I say, okay, let's feed in Ectocarpus siliculosus chart into that and kind of integrate it into the brown algae. And that's when I found there is a chat sequence in rice, and it fits itself very beautifully between the trypanosomes and the brown algae, just based on its the protein sequence domain similarities. And then it goes into anthracnose fungus. Both those fungi which you see right below the ectocarpus, these are the fungi which infect rice. I'm like, let me make a wild guess here. Maybe the chat jumped from a fungus infecting rice, and I don't know. Maybe before it became a neurotransmitter, it was a regulatory molecule which found its way into the realm of animal world. But I went back to look at the most distant ancestor. Again, it was a very, very complex phylogenetic tree. Just for the sake of the talk, I kind of refined it so that it gives you a clearer view. Now let's see. The ancestor I found is what is a very fascinating organism. It's called Ostriococcus. And it's let me see, I need to pull up the Ostriococcus. It belongs to what is called a Prasinophyceae, an early diverging class within the green plant lineage. This is a very fascinating organism. It's a single-celled alga. It thrives in the upper illuminated water column of the oceans. The most striking feature is how minimal it is. A minimal cellular organization. Naked, nearly 1 micron cell, doesn't have any flagella. And look at this. It has one single chloroplast and one single mitochondrion. 3 different ecotypes based on the adaptation to light intensity. So if you want to see a transitory organism where you want to look at photosynthetic processes, glycolytic pathway, and a non-neuronal acetylcholine, this would be an amazing organism to look at, right? So that's where I stepped out from the computer, and bamboo and rice, they both belong to the family called Poaceae. And since I now have a well-characterized chat sequence in rice, I presented my idea. I said, I will isolate the cDNA clones of chat and ACHE in bamboo. And bamboo seems to be a great organism system. The top portion of bamboo shoot has acetylcholine content of 2.9 millimolar per gram, 80 times of what is found in a rat brain. What I'm looking at is tissue cultures of bamboo, where I would perturb the acetylcholine levels. I have a bamboo genetic transformation system where I can alter the expression of choline acetyltransferase and acetylcholine esterase. And I will analyze those transgenic plants not just for the altered acetylcholine content, since altering the non-neuronal acetylcholine can perturb genes which are involved in cancer, dementia, Alzheimer's, what have you. That's when I said, okay, transgenic bamboo, where we alter. I mean, it's impossible to find a human volunteer where I can alter his non-neuronal acetylcholine, right? But a plant tissue culture will not complain, right? So can I look at those parallels? Just look at the genes. Just look at the genes. The genes may have a different role in plants, but can I illuminate the involvement of non-neuronal acetylcholine in the pathogenesis of diseases involving inflammation, local and systemic infection, tumorigenesis, where modification of cell structure and function play a key role? Tangential, yes, but we're looking at molecular basis of diseases using plants. And I was throwing this idea out. I went to NSF, I went to NIH, I went to the Kentucky Cabrin system and everybody was like, fascinating, fascinating. But I don't know whether we can fund it. I even went as far as to some private investors, and there was one guy who called me from DARPA. He said, wow, fascinating, fascinating. But in a boardroom of two guys voted it down to one guy who was fascinated. But that's when I put it back in the attic. But it's like, I was born on April 16th. I share my birthday with Charlie Chaplin. I was born a day later than Leonardo da Vinci. So Leonardo and Charlie Chaplin have a lot of projects which are in their attic. So yeah, a lot of unfinished stuff, right? But I was still teaching. And as if that was not enough, Rice had another surprise. One of my students, his name is Bobby Voiles. He's now a cardiologist. I would love for him to have become a neurologist, but I think money speaks, I guess. I don't know. But he came up and he said, I found something else, which is closer to your bamboo story. And this is about a gene called the warrior gene. What is this all about? It's a gene called monoamine oxidase type A. This study was done to examine a gene associated with MAOA. It's a chemical found in the brain. This chemical is associated with violence and aggression in humans. And the first research in the late 1970s was looking for MAOA as an antidepressant target for drug companies. Other research involved lower MAOA levels, causes an increase in happiness of women. So that was a paradox. The same gene which causes happiness in women is responsible for violence and aggression. That was a humorous paradox. I saw that. But there was a controversy. It was done, the research done in 2006 when a researcher linked MAOA to the heightened levels of aggression in the Maori people of New Zealand, right? Something we see in the New Zealand parliament nowadays a lot. That controversy came about because the researcher called it the warrior gene. I mean, that was almost like a racial slur. But that was what the warrior gene is all about. So what Bobby, my student, saw was he just blasted MAOA, and this is what he found. I just am representing the printout which he got to me very excitedly, right? He said, it starts with macaca, mulata, and Homo sapiens in one branch of the phylogenetic tree. Then it grows down to a domestic ferret, and then a vampire bat, and a Norway rat, and then oyster, tick, protobacteria, anthrax, lice, fluke. But he said, look at this. There is a branch which has grape, rapeseed, and Japanese rice. And I'm like, wow, man, is this another parallel? So what do we have here? A testable hypothesis again, warrior gene in a rice ball, one of the most important organism where we found a related sequence. And this is not like, it's almost like a 70 to 80% similarity. So now rice can be a non-human model to test for the molecular basis of a warrior gene, a gene which is involved in violence and aggression. We have a wonderful tissue culture and transformation system in rice. So can we manipulate? And this is way back in time. Now with CRISPR, I don't know what else can we do. Parallelly, I was doing to complete the stir fry, bamboo, basil, rice, and now basil, right? Just to add some tinge to my stir fry, right? This is when I was looking at basil, and it has a compound called eugenol. I was testing plant extracts on human cell cultures, especially cancer cell. And I found eugenol to be a phenylpropene, a plant volatile compound, which actually has multiple roles, defense and pollinator attraction, but it has a propenyl side chain, and eugenol synthase catalyzes technically the reductive displacement of acetate from the propenyl side, which produces this compound, this fascinating compound called eugenol. And when you pour eugenol on a cancer cell culture, within 72 hours, it kills the tumor. So I said, okay, now I'm not letting it go. I'm looking at eugenol synthase bioinformatics. And I found that it has a domain called NMRA, a transcriptional regulator, which is a system controlling nitrogen metabolite repression in fungi. Nitrogen metabolism is an important factor during chemotherapy of cancer. When I looked at it, the last ancestor was a black mold. And we already have a very good transformation system, which we developed in eugenol parallelly. So now we have 3 different tissue culture transgenic systems: bamboo, basil, and rice. And I said these can be potential non-human models to explore molecular basis of cancer. Can we observe certain molecular perturbances which will actually inform us that there are genes which are crucial to cause inflammation, and that can be extended to know the molecular basis of cancer, dementia, atherosclerosis. Am I blowing hot air here? Plants to humans that jump? No, sir. I projected this slide once when I was lecturing people on how eureka moments in one system can cause applications in another system. This is petunia. I call the lecture Petunia for Human Health. On the right, I project a flower petunia. On the left, I show a tumor, an arthritic hand, an Alzheimer's patient, an asthma patient, a slide of leukemia, and I said, you want to see what's going on here, right? Let me tell you the story of Richard Jorgensen. He used to work in a small company called DNA Plant Technology Corporation, 1999. He wanted to make bright petunias. So what did he do? He took that RNA which causes the anthocyanin pathway, if I'm not mistaken, and he wanted to put it in, saying that, okay, more of the same. But he got a white petunia. He got a variegated petunia. And he accidentally discovered a process called co-suppression. When he dug back in time in 1928, in a very obscure journal called the Journal of Agricultural Research, obscure for the scientific community, but not for the agronomists, at the Virginia Agricultural Experimental Station, a person by the name S.A. Wingard saw a strange occurrence. A virus attacks tobacco plants. The leaves become bad, but the new leaves are all fine. So much so that the Farmers used to take those affected leaves and rub them on all tobacco plants. What are they doing? They are using a diamond to cut diamond. That's when this process called RNAi came up. But all this research was in plants. But in comes Andrew Fire and Craig Mello from Stanford and the University of Massachusetts, Nobel laureates, 2006. They worked out those essential steps of RNAi, what was learned in plants, and they synthesized what is called the siRNA, the short interfering RNA, to silence target genes in cells. RNAi left plants to get into the mammalian systems. Gregory Hannon at Cold Spring Harbor actually found out that you could send in your target gene. There is a compound called dicer in your system which chops that gene, synthetic or natural, and it gets into what are called argonaut proteins. Argonaut as in Jason and Argonauts going in search of the golden fleece. Only here, that short interfering RNA will go to a target. This is a precursor of the CRISPR mechanism, right? It goes there and it chops off whatever you want. And what happened then? We have a treatment for HIV, which was born from this. You go and you get a healthy HIV positive, chemo responsive AIDS lymphoma patient. We go through a process called apheresis and enrich blood, which contains stem cells, which can go into a lentivirus, and you go to an AIDS patient and you chop off the AIDS. Just like what Richard Jorgensen observed in Petunia. Petunia was a model system. Then the model system jumps to C. elegans when those Nobel laureates came. So what you learn in plants is now treating AIDS patients. So what am I saying? Rice can be a model research plant, a non-human model for examining molecular therapeutics. Bamboo will be a new addition as a non-human plant model. Basil will enhance its status from being a phytopharmaceutical to an experimental non-human model. Right. The stir fry is actually, if you look in the East, China especially, if you go to a Chinese restaurant and you order a dish called Kung Pao chicken, it contains bamboo shoots along with peanuts and with a generous helping of basil leaves. And on the side you have rice. So my bamboo, basil and rice, and they might be eating all the acetylcholine in the world, right? Tender bamboo shoots. That's why there is very, very less incidence of cardiac cancer diseases in the East. There was a fascinating study called the China study that was done. It's all dietary. They might be actually perturbing those non-neuronal acetylcholine levels. A very wild hypothesis from my side, but something that cannot be ignored. Right. So that's my attic science proposal. If it has kindled certain brains in the audience, I would be very, very gratified to see if I can take this forward. I would love to pull it off my attic someday, right? Because they say it's not the fruits of scientific research that elevate a man and enrich his nature, but the urge to understand. That's what Albert Einstein said. We need to continue to understand life. I would like to close by another amazing passage I want to read out to you, because it's Memorial Day, and amongst my friends, they might be ex-servicemen or still people active in service. I want to salute them. I want to thank them. But whenever I think of Memorial Day, I'm reminded of a poem that was written by W.B. Yeats. It's called An Irish Airman Foresees His Death. Bear with me, because this is one of my favorite poems. And it has a deep connection to what I presented to today. And I'll explain that connection. Well, the poem reads, I know that I shall meet my fate somewhere among the clouds above. Those that I fight, I do not hate. Those that I guard, I do not love. My country is Kiltartan Cross, my countrymen Kiltartan Spur. No likely end could bring them loss or leave them happier than before. But here comes the most beautiful lines ever. Nor law nor duty bade me fight, nor public men nor cheering crowds. And here goes. A lonely impulse of delight, a lonely impulse of delight drove to this tumult in the clouds. I balanced all, brought all to mind. The years to come seemed a waste of breath. A waste of breath the years behind, in balance with this life, this death. What I live by is this, the lonely impulse of delight. And that lonely impulse of delight gives rise to what a amazing thing science is. You hold on to certain ideas which may be crazy, but as they say, somebody said, you need to stay hungry and stay foolish. So I wish you all a very happy Memorial Day, but I thank the servicemen because they are there because of this lonely impulse of delight, and that's what we need to learn from them. They keep us safe so that we can have great ideas, right? Thank you all. I can answer some questions. Thank you so much. Thank you so much. Thank you so much. Yeah, one interesting question is belladonna is a natural anticholinergic in humans, but neural anti-acetylcholine, I believe. So maybe this plant evolved to make this effect in humans. Yeah, that's an amazing thought. And to think of it, I mean, my company here, we work with various crops. I work with lime, peanuts. We also work with cannabis. We have a cannabinoid system inside us, which is so receptive to the cannabinoid pathway. And apart from all the recreational uses of cannabis, it's amazingly the medicinal stuff found in that plant. It treats neurological diseases. Yes. Belladonna, yes. At one time when I was a kid, there was a brain fever epidemic, right? That used to happen a lot in India. And belladonna was one potent thing which kind of eradicated that endemic at that time, right? Well, does my work provide any, Sis is asking me this question, any insights into why very large animals like elephants and whales are very resistant to cancer? That's a very, very interesting thing to look at. If you look at elephants and whales, they have an amazing memory power. And that again points to the role of acetylcholine, both neuronal and not neuronal. And the other thing is, elephants, they live long. Again, basically, living long and having an excellent memory, again, looks at the acetylcholine pathway, because it's involved in cellular organization, cytoskeletal framework. So maybe we are looking at a resistance to cancer, again, to be noted to looking at this cholinergic, non-cholinergic pathway, right? So, yeah. So belladonna, yes, can be a very beneficial compound. There is treasures to be mined in the plant world, right? So cancer grows much faster than zero G. Not totally unrelated. In fact, it's not just cancer. They sent out plant cell cultures into space, into the International Space Station. And the division of those plant callus cultures or suspension cultures was huge in zero G. So now we have to look at, this is an amazing thing. When we look at the influence of sound waves, like there are some vibratory systems, we have some sonicators. If we put the plant cell cultures in there, the division is somehow, like even a simple vertex of those plant cells makes them grow faster. Some of the recalcitrant species I observed, when I was doing my transformation experiments with callus cultures, if I sonicate or if I vibrate them, the cells divide much faster. So yeah, something like that might be happening in zero G. And again, these are things which come in the realm of crazy science, but someday, yeah. So cancer grows the same in 10 days yeah, as opposed to 10 years on Earth. Yeah, recent discovery. That's an amazing point to share. It's so yeah, cellular organization. We need to stop looking at these diseases as diseases, because everything starts with inflammation, right? So inflammation needs to be looked at, but more importantly, not just cancer cells. Max, I take that point. Not just cancer cells, but every cell, because every cell is a cancer cell waiting to happen. I remember reading this book called One Renegade Cell. We have all these cells, right? 34 trillion cells inside our body. It's like I read somewhere that when you look at the cells inside the body, it's like every 3 days your stomach lining is replaced. You wake up every day with almost 90% of your pancreas being replaced, right? Every 7 years of your life, every one of those 34 trillion cells is being replaced by a new cell. 7, 14, 28. Look at that cycle. And the people who live longer are the ones who duck detox, who are replacing those cells. But what do? We go out, have black coffee, have chocolates, have beer. And yeah. So we are slowly, the human body is actually programmed to live for 195 years, but we just have our coffee, our beer, and breathe that wonderful air around nowadays. That's what causes all the things, right? So, yeah. So that's another thing. Cells need to be looked at as cells, because that cancer cell is one renegade cell which rebels, the spoiled child. But if you look at what happens at the molecular level, I think that's something to look at. So, and when we can't have human volunteers, we have plants. They don't complain, right? Someday we might know more of their feelings, but what plants feel now, yeah, we can look at the molecular system, look for those common genes. Like there is a P53 gene which is involved in cancer, which has an exact replicate in the corn. Like Barbara McClintock looked at transposons in corn, maize, and the transposable element biology which she worked out in maize plant is now being used to treat what is called the bubble boy disease. That's why her Nobel Prize, because there is no Nobel Prize in plant biology, she gets her Nobel Prize in physiology and medicine. But all her life she worked with corn. So there you go. So I'm not too far off when I say plants have been wonderful model systems to solve medical problems. Barbara McClintock's Nobel Prize in Physiology and Medicine is a case in question where a plant scientist gets a Nobel in medicine and physiology, right? So, yeah. So that's what my attic science is all about. There are many more, but I'll have other occasions, I'm sure, where I can again share with you some crazy science. But this is one of my favorites. Looking at how a neurotransmitter that flows all through our body might have jumped from another realm of life into us, and it somehow programmed itself to memory and cognition from cellular organization and phytochemical pathways. Well, thank you guys. If there are no more questions, then we shared a memory today. So I'm glad I did. Thank you. You guys all have a wonderful weekend, and keep those lonely impulses of delight coming. All right. Thank you. Thanks, Stephen. We'll see you soon. Bye.